In this module, we are going to talk about microbial control. We know that bacteria do not need much to grow and survive. A good carbon source, a nitrogen source, and water are mainly what they require. Hence, they grow almost everywhere in all those places where we would actually prefer them not to grow. They grow on and in our bodies and cause diseases, destroy crops by infecting them with diseases, bring about food spoilage, contaminate our pure cultures, and can be, in fact, a source of nuisance to us. So we are constantly in need of methods to keep these microbes at bay and to control them. Equipped with our knowledge of microbial growth, it is easier to control microbes. So we'll focus now on the different methods of microbial control. Controlling microbes can mean killing the microbes present or inhibiting their growth. Those substances which kill microbes are called bactericidal agents, and those which inhibit their growth are called bacteriostatic agents. When we are targeting at controlling microbes, we mainly are interested in cell death. But the cell death in a population does not occur instantly. Instead, just as cells grow exponentially, they also die exponentially. Whatever treatment the bacteria are subjected to, they die at an exponential rate. Exponential death means that they die at some fractional rate per unit time. If 50% of the microorganisms in a population die every minute, then after 2 minutes, there will be 25% still alive. After 3 minutes, there will be 12.5% still alive. And after 4 minutes, there will be 6.25% still alive, and so on. If the logarithm of the population number remaining is plotted against time of exposure of the microorganisms to the agent, a straight line will result, as can be seen in the figure on your screen. The death of a given microbial culture depends upon a few factors which have to be considered while selecting the control method. The first factor is the degree of contamination, which is also called the microbial load. The more the microbial population, the more time it will take for sterilization. A high population load will require more time as compared to a low population load as shown in this figure. The second factor is the susceptibility of the microbes to the antimicrobial agents. It is necessary to carefully select an antimicrobial agent because some microbes are resistant or less susceptible to certain antimicrobial agents. For example, it is no use trying to kill Deinococcus radiodurans using radiations because it is resistant to radiations. The third factor is the time of exposure. Time is a factor relative to other factors. More the microbial load, more is the time required for killing the entire bacterial population. Also, more the concentration or intensity of the antimicrobial agent, less is the time required. For example, when 1% phenol is used as antimicrobial agent, it takes 20 to 30 minutes to kill the bacterial population, but when 5% phenol is used, it takes just 2 to 3 minutes. The next factor is concentration or intensity. The concentration of the chemical agent and the intensity of the physical agent determines the time required to kill a microbial population. For example, we just saw that 5% phenol takes lesser time to kill the microbial population as compared to 1% phenol. But sometimes the case is just reverse. Like 70% ethanol is more effective than 100% absolute ethanol because the presence of water enhances the activity of alcohol. The next factor is physiological condition of microbial population. The effect of the antimicrobial agent depends upon the physiological state of the microbial population. Young, actively growing cells are killed more easily than old, dormant, non-dividing cells. The next factor is the type of the microorganism. The effect of the antimicrobial agent differs with the type of the microbes like bacteria, fungi, viruses, etc. The activity differs for gram-negative and gram-positive bacteria within a group too. Some species are more resistant to antimicrobial agents than other bacteria. Furthermore, bacterial as well as fungal spores are more resistant than the vegetative cells. The next factor is environmental factor. Many environmental conditions like temperature and pH influence the effect of antimicrobial agents. For example, the killing action of ethanol is greater at higher temperature than at lower temperature. Also, the presence of organic material like dirt, oil, saliva, blood, fat, etc. might interfere with the penetration of heat, chemicals and radiations and also inactivate the chemical disinfectants. Let us first discuss some commonly used terms in microbial control. The first term is sterilization. It is defined as the process where all the living microorganisms, including bacterial spores, are killed. There is no degree of sterilization. An object is either sterile or not. Sterilization can be achieved by physical, chemical, and physicochemical means. The second term is disinfection. Disinfection is the process of elimination of most pathogenic microorganisms on inanimate objects. Disinfection can be achieved by physical or chemical methods. Chemicals used in disinfection are called disinfectants. Different disinfectants have different target ranges. Not all disinfectants can kill all microorganisms. The next term is decontamination. It is the process of removal of contaminating pathogenic microorganisms from the articles by a process of sterilization or disinfection. It is the use of physical or chemical means to remove, inactivate or destroy living organisms on a surface so that microbes are no longer infectious. The next term is sanitization. Sanitization is the process of chemical or mechanical cleansing applicable in public health systems, usually by the food industry. The next term is asepsis. Asepsis is the employment of techniques to achieve microbe-free environment. Antisepsis is the use of chemicals on skin or mucous membranes to keep it free of pathogenic microorganisms. An antiseptic is a substance that prevents the growth and reproduction of various microorganisms on the external surfaces of the body. Bacteriostasis is a condition where multiplication of the bacteria is inhibited without actually killing them. Bactericidal is that chemical that can kill or inactivate bacteria. Such chemicals may be called variously depending on the spectrum of activity such as bactericidal, virucidal, fungicidal, microbicidal, etc. Let's now talk about the common methods of microbial control. We'll be talking about the physical agents, which would include heat, radiations, and filtration. We'll also be talking about chemical agents, which will include phenols, alcohols, halogens, and gaseous agents in the coming slides. Moving ahead, let us talk about the physical methods of control. The physical methods of microbial control include temperature, irradiation, filtration, dehydration, etc. Let us first talk about temperature. We already know the influence of temperature on the growth of microbes and how every organism has its own optimum growth temperature. Naturally, temperature above the optimum would be lethal and can be used to kill the microbes. So also, temperatures below optimum can be used to retard the growth of microbes. High temperature is the most widely used and reliable method of sterilization, including involving destruction of enzymes and other essential cell constituents. The efficiency with which high temperature is able to inactivate microorganisms is dependent upon the degree of heat, the exposure of time, and the presence of water. The action of heat will be due to the induction of lethal chemical events mediated through the action of water and oxygen. In the presence of water, much lower temperature time exposures are required to kill microbes than in the absence of water. Both dry heat and moist heat are used for sterilization. When trying to develop an effective heat treatment for a food or culture medium, it is often necessary to quantitatively determine the rate of death. Initially, thermal death point was used to express this. It was defined as the lowest temperature at which the bacterial population is killed in 10 minutes. This was then replaced by the concept of thermal death time. The thermal death time is the time it takes for a population of cells to reach effectively zero at a given temperature. But it is virtually impossible to reach zero value. Hence, decimal reduction time or D value is more accepted these days. The decimal reduction time is a measure of heat sensitivity of a microbe and is defined as the length of time it takes for the viable population to decrease tenfold or 1 log cycle at a given temperature. Hence, the time or the dose it takes to reduce 1000 microbial cells to 100 cells is the T value. This can be determined by incubating a culture at a lethal temperature and periodically determining the number of surviving microbes in the sample by viable plate count. This is called the survival curve method. The log of the viable plate count is plotted against time and a straight line is drawn through these points. The negative reciprocal of the slope of the line is the decimal reduction time. The value is only valid for the microbe under study at that temperature and in the medium used. For example, the D value at 72 degrees of 1 minute means that for each minute of processing at 72 degrees, the bacterial population of the target microorganisms will be reduced 90%. In this figure, the D value is 14 minutes, 40 minus 26, and would be representative of a process at 72 degrees. Once D values have been determined for at least 3 temperatures, the Z value can be determined. The Z value measures the sensitivity of a microbe to a change in temperature and is defined as the amount of temperature increase necessary to decrease the D value tenfold. To determine the Z value for a microbe, at least 3 D values are plotted against its temperature and a line is drawn through the points. The Z value is the negative reciprocal of the slope of the line. With a Z value in hand, it is possible to predict the microbe's death rate in the specific medium at any lethal temperature. D values, TDT and Z values are very useful in the food industry where one wants the treatment to appropriately reduce the microbial population with minimum negative effect on the food itself. Let us now talk about the types of heat treatment. Dry heat. The first is flaming. This is one of the most ancient methods of killing microorganisms and is commonly used in laboratories nowadays. Here the article to be sterilized is passed over a Bunsen burner's flame but not heated to redness. Articles such as scalpels, inoculating loops, mouth of test tubes, flask, glass slides, etc. are passed through a flame a few times. Even though most of the vegetative cells are killed, there is no guarantee that spores too would die. In this figure, you can see the flaming of an inoculating loop. The next method of dry heat is hot air oven. Dry heat is sometimes recommended for sterilization where the moist heat can spoil the materials like oils, powders, etc. The apparatus used is a hot air oven and can be routinely used for sterilizing glasswares like petri plates, pipettes, etc. A 1 to 2 hour exposure at 160 to 180 degrees is enough to bring about sterilization. Since air is a poor conductor of heat, even distribution of heat throughout the chamber is achieved by a fan. The heat is transferred to the articles by radiation, conduction, and convection. The oven should be fitted with a thermostat control, temperature indicator, meshed shelves, and must have adequate insulation. Articles to be sterilized must be placed at sufficient distance so as to allow free circulation of air in between. Mouths of flasks, test tubes, and both ends of pipettes must be plugged with cotton wool. Dry heat is an effective method of sterilization of heat-stable articles. The articles remain dry after sterilization. This is the only method of sterilization of oils and powders. However, there are a few disadvantages. These are, since air is a poor conductor of heat, hot air has poor penetration. Cotton wool and paper may get slightly charred, glassware may become smoky, and it takes longer time compared to autoclave. Let us now move on to moist heat. Boiling is the most common method. Boiling water kills most vegetative bacteria and viruses immediately, but some bacterial spores are resistant to boiling and survive. Hence, this is not a substitute for sterilization. Certain bacterial toxins such as staphylococcal enterotoxin are also heat resistant. The killing activity can be enhanced by addition of 2% sodium bicarbonate. When absolute sterility is not required, certain metal articles and glasswares can be disinfected by placing them in boiling water for 10 to 20 minutes. Before the advent of the disposable plastic syringes in hospitals and dispensaries, the glass syringes and needles were disinfected by boiling. Boiling is also used as a household method of disinfecting milk bottles of infants. The second method of using moist heat is Arnold steam sterilizer. The Arnold steam sterilizer was widely used in US and is based on the principle of live steam. As shown in the figure, it consists of a cylinder of tin or copper with a cover, which is enclosed in a movable cylindrical outer cover or hood. The inner cylinder has a perforated bottom, as you can see, through which the steam may enter. The steam comes from the small chamber below the copper bottom, which contains water which is boiled. The peculiarity of this type of sterilizer is that the steam which escapes from the sterilizing chamber condenses beneath the outer cover and falls back upon the chamber in which the steam is generated. Thus, this sterilizer uses live steam which has a temperature of 100 degrees and a single exposure for 90 minutes can satisfactorily sterilize the materials. The next method is tyndallization. Certain spore-forming bacteria can resist the action of steam for hours. Such prolonged exposure to steam might be harmful to the culture media containing gelatin, milk and sugars. Hence, fractional or discontinuous sterilization or tyndallization is used. Here the medium is exposed to steam for 3 consecutive days. The idea behind it, as proposed by John Tyndall, was that the culture medium to be sterilized might contain bacteria in their vegetative stage as well as in the spore stage. So the media to be sterilized is boiled for a period of time, say 30 minutes, and then cooled for 24 hours and then boiled again. This boiling is repeated 3 times. What is achieved during the period of intermittent cooling? Boiling is enough to kill vegetative cells, but not the spores. But during boiling, the spores are activated, and during the cooling period, they germinate to form vegetative cells, which are in turn destroyed by the next boiling phase. The most common method in every microbiology lab for using moist heat, however, is autoclave. It is the most practical method of sterilization. The autoclave was invented by Charles Chamberlain in 1884. It is a closed device which works on the principle of saturated steam under pressure, the same as that of the pressure cooker. It uses moist heat to kill not only all the vegetative cells but also the endospores. Moist heat is more effective than dry heat because it has more penetrating power. It is supposed to act by degrading nucleic acids and denaturing proteins. At a pressure of 15 pounds inside the autoclave, the temperature is said to be at 121 degrees. Exposure of articles to this temperature for 15 minutes is enough to bring about sterilization. Let us now talk about the construction and working of an autoclave. A simple autoclave has a vertical or horizontal cylindrical body with heating elements, a perforated tray to keep the articles, a lid that can be fastened by screw clamps, a pressure gauge, a safety valve, and a discharge tap. The articles to be sterilized must not be tightly packed. The screw caps and cotton plugs must be loosely fitted. The lid is closed but the discharge tap is kept open and the water is heated. As the water starts boiling, the stream drives air out of the discharge tap. When all the air is displaced and the steam starts appearing through the discharge tap, the tap is then closed. The pressure inside is allowed to rise up to 15 pounds per square inch. At this pressure, the articles are held for 20 minutes, after which the heating is stopped and the autoclave is allowed to cool. Once the pressure gauge shows the pressure equal to that of the atmospheric pressure, the discharge tap is opened to let the air in. The lid is then opened and articles are then removed. There are certain precautions which have to be taken while using an autoclave. Firstly, the autoclave should not be tightly overpacked because steam needs to circulate freely in the chamber and contact each and every object. Secondly, if the chamber is not totally free of air, then it will not reach a temperature of 121 degrees even if it shows a pressure of 15 pounds. And remember, it is not the pressure which kills the microbes but the high temperature which is achieved by steam under pressure. Thirdly, when the pressure is released, it should be done slowly or else the contents inside will be thrown upwards. Also, since moist heat is used, heat labile objects cannot be sterilized using an autoclave. There are certain chemical, biological and physical indicators that can be used to ensure that an autoclave has operated for the prescribed time at the correct temperature. Chemical indicators consist of a sterile autoclave tape which changes color once the correct conditions have been met. This indicates that the object inside the package or under the tape has been autoclaved sufficiently. Biological indicators include a culture tube containing sterile medium and spores of heat-resistant bacterium Bacillus stereothermophilus. If the autoclave did not reach the right temperature, the spores will germinate and their metabolism will change the color of a pH sensitive chemical. Physical indicators often consist of an alloy designed to melt only after being subjected to 121 degrees for 15 to 20 minutes. If the metal has melted, the change will be visible. Moving ahead to pasteurization. This is a process whereby milk and food are held at temperatures below their boiling points for a specified time for killing harmful organisms such as bacteria, viruses, protozoa, molds, and yeast. The process was named after its inventor, French scientist Louis Pasteur. Milk pasteurization. France-born Soxhlet in 1886 first adapted pasteurization for preserving milk. Two methods are very commonly used to pasteurize milk. High temperature short time method. It's also called flash pasteurization and is by far the most common method. It involves holding the milk at temperature of 72 degrees for at least 15 seconds. The high temperature short time pasteurization standard was designed to achieve a 5 log reduction in the number of the viable microorganisms in milk. This is considered adequate for destroying almost all yeast, mold, and common spoilage bacteria, and also to ensure adequate destruction of common pathogenic heat-resistant organisms, particularly Microbacterium tuberculosis, which causes tuberculosis, and Coxiella burnetii, which causes Q fever. The next method is ultra high temperature, which involves holding milk at a temperature of 138 degrees for 2 seconds. Ultra high temperature treated milk does not require refrigeration and can be safely stored at room temperature. The next physical method of microbial control is irradiation. Radiations are mainly of two types. Ionizing radiations. These have enough energy that they can act on the target molecules, remove electrons and form ions. X-rays, gamma rays, and electron beams are examples of ionizing radiations. Their wavelengths are smaller than 1 nanometer. The second type of radiations are non-ionizing radiations. These cannot form ions. UV rays are a type of non-ionizing radiations. Their wavelength is longer than 1 nanometer and they are less effective as compared to ionizing radiations. Ionizing radiations like X-rays and gamma rays collide with various molecules in the microbes and produce hydroxyl and hydride radicals. These highly reactive molecules are capable of degrading DNA and proteins and hence the subsequent death of the microbial cells. UV radiations bring about damage in the DNA. They cause the formation of covalent bonds, particularly two adjacent thymines will undergo dimerization to form what is called as thymine dimer. The two thymines undergoes bond formation between the fifth and the sixth positions to form what is called a cyclobutyl ring. The formation of the dimers causes a distortion in the DNA strand and prevents its replication and transcription and brings about the death of the cell unless the damage is repaired. The amount of radiation necessary to kill a microbe is species specific and depends on a variety of factors. Gram-negative bacteria like Pseudomonas, Escherichia, Shigella and Salmonella are more sensitive to radiation than most gram-positive bacteria. Spores of Bacillus and Clostridium are even more resistant due to special proteins in the spores that are thought to protect the DNA. The use of radiations for sterilization is measured in grays. 1 gray is defined as absorption of 1 joule of radiation per kilogram. In general, microorganisms are more resistant to ionizing radiations than higher organisms. For example, the amount of energy required to reduce the bacterial load by 10-fold is at least 200 grays, whereas the lethal dose for humans is just 10 grays. Radiation is currently used as a sterilization practice in food and drug industries. Food and Drug Administration has approved the use of radiation for sterilization of surgical supplies, disposable labware, drugs, and even tissue grafts. The third physical method of microbial control is filtration. Though heat is the most common and easy method of sterilization, not all materials can be sterilized by it. Heat-sensitive liquids like hormones, vitamins, etc. and gases are sterilized by filter sterilization. Filtration physically removes the microbes by employing a filter or a membrane which has pores too small to allow the passage of microbes but large enough to allow the passage of liquid or gas. We know of microbes which have a diameter as small as 0.3 micrometers. Hence, the pore size of the membranes commonly used for microbiological purposes is 0.2 micrometers. But a filter of this pore size would not remove viruses because they are smaller in size. A filter system with sufficiently small pores to remove viruses would have an extremely slow flow rate due to the viscosity of water. Because viruses are not removed, filtration cannot be technically considered a form of sterilization, although it is common to refer to the process as filter sterilization. Let's talk about the types of filters. There are 3 major types of filters. The first are what are called as depth filters, which are the oldest type and are made of overlapping layers of fibrous sheets of papers, asbestos or glass fibers. This type of filters are mainly used as pre-filters and they efficiently remove the larger particles so that they do not clog up the more sophisticated filters during the final filter sterilization process. The most common type of filtration method used in modern microbiology labs are membrane filters. These are made from high tensile strength polymers of cellulose acetate, cellulose nitrate, polycarbonate, polyester, polypropylene or polysulfone. These polymers appear as a thick mesh of interconnected strands with rather precisely defined spaces between the polymers. The third type of filters are nucleopore filters. These filters are created by exposing a very thin polycarbonate film to nuclear radiation, which creates areas of local damages in the polymers. The filters are held in special holders or holding apparatus of glass or metal. The solution to be filtered is forced through the filter with the help of a vacuum or pressure using a syringe or a peristaltic valve. The filtered solution is collected in a previously sterilized container. The filtering apparatus and filters are generally sterilized separately and assembled aseptically, or they are available pre-sterile.